A very good morning to all. I'll be discussing today on interplay of microbes in hepatobiliary malignancies. So there were few questions that I needed to answer. That is there any difference in the gut microbiome of HCC versus cirrhosis or HBV and HCV associated cirrhosis? How do these microbes contribute to the pathogenesis of HCC or the cholangiocarcinomas? And do these microbes manifest as clinical significant infections? And are these infections associated with the treatment modalities as well? And how to manage these infections? So changes in the gut microbiota composition associated with HCC have been studied widely in the animal model, and various animal models where the dimethylbenzene have been used, or dinitrosyl ethylamine induced HCC, and they have seen that there is altered gut microbiota. Also, there is increase in species like Tropobium bacteroides, Bacteroides veligeris, Bacteroides acidofaciens, and also certain Clostridial species. Few studies have shown that E. coli, Tropobium, also various species of Tropobium have increased. However, Lactobacilli, Bifidobacterium, and Enterococcus species have reduced diversity in these mice. Thus, it has been proven that in mice, the gut microbial diversity is definitely altered in case of HCC. Also, there are various human studies which show that the gut microbial composition changes in the HCC, and there is increased pathogenic bacteria like E. coli, Proteobacteria, Enterobacter, Prevotella species which are increased. And however, certain other species like Akkermansia and Bifidobacterium, which are the healthy gut bacteria, are reduced. So, there is in this study where the gut microbiome in HCC versus cirrhosis and healthy controls was studied, and early HCC patients, 75 patients versus the patients with cirrhosis were compared to the healthy controls. And here they observed, this is the control arm and this is the liver cirrhosis with compared to the early hepatocellular carcinoma. So here, compared to the controls, the fecal microbial diversity was significantly decreased in cirrhosis. However, in contrast, the microbial diversity was increased as compared to cirrhosis in case of HCC patients. So fecal microbial diversity was decreased from healthy control to the cirrhosis, but was increased from cirrhosis to HCC, early HCC, as compared to cirrhosis. So does this have a beneficial effect? So the answer is clearly no, because such increase in microbial diversity has also been observed in adenocarcinoma of the intestine. And here, when the cycle of the malignancy transforms from adenoma to an adenocarcinoma in the intestine, the microbial diversity increased. However, it was not the beneficial bacteria that were increasing. Similarly, here in HCC also, though there is increase in the microbial diversity, it is not beneficial because the harmful bacteria or the bacteria which are producing lipopolysaccharides are increasing and there is overgrowth of such bacteria, which is leading to the increased diversity. So butyrate producing Ruminococcus, Oscillobacter, Faecalibacterium and Clostridium are usually reduced, and lipopolysaccharide producing Klebsiella, Haemophilus are increased as compared to the controls. So what is the effect of this, we'll be seeing in subsequent slides. Butyrate is considered as an important regulator of gene expression, inflammation, differentiation and apoptosis in host cells, and it also plays a key role in bacterial energy metabolism and gut health. Decrease in such butyrate producing bacteria may promote intestinal destruction and thereby contributing to the HCC development. Certain studies have also studied the role of microbial diversity from NAFLD and HCC. So beneficial indigenous bacteria such as Lactobacilli, Bifidobacterium and Bacteroides species are decreased, while potentially pathogenic bacteria such as Proteobacteria, Fusobacterium, Vallionelaceae and Streptococci, that is Enterococcus family, they are usually increased in HCC. Also, HBV associated cirrhosis versus HCC, various studies have shown that the gut microbiome characterization on different independent cohorts of control chronic hepatitis B patients, liver cirrhosis and then HCC patients. Here they have shown that the gut microbiota diversity is different in each subgroup of the patients, and again in this study also Coprococcus and Faecalibacterium were depleted in HCC. Similar differences have also been seen in the cohorts of HCV associated cirrhosis and HCC, where there is a reduction in the ruminococcus and lacnospiracy in such patients. So how does this gut microbial diversity or dysbiosis affect the development of HCC or carcinogenesis? How is this promoted? So there are two main factors, that is dysbiosis and the leaky gut, and then there are microbial metabolites which are predominantly important in the pathogenesis of HCC in such patients. So there are various molecules like LPS producing bacteria, as we have seen, are increased in HCC. So LPS can cause inflammation through the TLR4 and is most commonly used as a marker of the microbiota associated molecular patterns. Intestinal bacterial metabolites usually regulate intestinal permeability and inflammation through the toll-like receptor 4 mediated pathways. Now these TLR4 pathways are important in HCC cells, and they have dramatically improved invasion and migratory capabilities as well. And TLR4 signaling induced leaky guts may enhance HCC progression. Another metabolite, that is calprotectin, which is usually mainly derived from the neutrophils, and it is a marker of inflammation. So calprotectin is produced in various body fluids, and it indicates inflammatory responses. As I've already mentioned, there was lower percentage of Akkermansia and Bifidobacterium detected in the feces of HCC patients. However, same patient had higher calprotectin levels in their stool, which was significantly increased, which represents more intense inflammatory response and hepatic injury that was ongoing in such patients. So how do these dysbiosis affects the gut microbial milieu in the HCC? So it is through the gut microbial metabolites. Now what happens is that through the portal venous circulation, gut derived metabolites, various cellular components, hormones, other substances enter into the liver and interact with the immune cells. However, these dysbiotic bacterial metabolites, they contribute to inflammation, hepatitis, cirrhosis, fibrosis, and even cancer, while others usually maintain the barrier integrity and prevent pathogen penetration. Short chain fatty acids, lactic acid, and the bile acids are the main microbial metabolites that have been studied. So let's have a look at short chain fatty acids. Now short chain fatty acids mainly that have a role are acetic acid, propionic acid, and butyric acid. These short chain fatty acids, these are main microbial metabolites of dietary fibers in the intestine and can suppress the hepatic inflammation by inducing regulatory T cells. Now in human beings, the maintaining the normal operation of the large intestine and morphology and function of the colonic epithelial cells depends on these short chain fatty acids. Now they promote the Treg cells during active immune responses and control cancer progression. So short chain fatty acids are beneficial. They will reduce the cancer progression by down-regulating the cancer-related pathways. And it has also been shown in mice studies that the HBx transgenic mice, that is hepatitis B virus induced HCC mice, have shown that the normal liver tissue development was seen and inhibited HCC growth when the short chain fatty acids were given to these mice. So thus, as we can see, dietary fiber usually in the presence of intestinal bacteria, they produce the butyrate. In the normal enterocyte, they usually lead to the decreased inflammation and other pathways that cause the healthy gut environment. However, in the cancerous cells, these usually lead to increased apoptosis and inflammation and decreased proliferation of the cancerous cells. These short chain fatty acids also determine the response to the therapy in HCC patients. Like if the patient is undergoing chemotherapy and he is a responder, the short chain fatty acid producing bacteria are increased and increased plasma level of butyrate are also seen. There is increased recruitment of CD4 and CD8 T cells and also increased levels of acetylated histones and decreased deacetylated histones that are seen, and increase in TNF alpha. This leads to increased apoptosis and sensitivity to the chemotherapeutic agent, decreased cell growth and proliferation, and also decrease in the migration and invasion caused by the cancerous cells. So thus response to the therapy is also dependent on the short chain fatty acids. Similarly, for immunotherapy patients, in the responders, increased butyric acid producing bacteria and increased fecal and plasma short chain fatty acids are observed, with increased inflammatory cells like CD4 and CD8 T cells, with decrease in tumor growth.
And use your pointer. Okay. We do not know where you are on this big slide.
So in immunotherapy patients also, the responders usually have increased butyrate producing bacteria and increased fecal and plasma short chain fatty acids. There is increased recruitment of CD8 and CD T cells that is seen, increased interferon gamma and TNF alpha, leading to decreased tumor growth increase tumor response to the immunotherapy and increase efficacy. So short chain fatty acids are beneficial in immunotherapy patients as well. Also radiotherapy responders have similar effects that increase butyrate producing bacteria and increase fecal short chain fatty acids. Decrease in interferon, decrease in antigen presenting cells and CD8 activation, decrease anti-tumor response. Ultimately leads to, if there is a high fiber diet and presence of short chain fatty acids in such patients, there will be reduced tumor growth, increased histone acetylation and increased radiosensitivity. So a patient with high fiber diet and presence of short chain fatty acids definitely increases radiosensitivity when they are treated. Another metabolite is lactic acid. It is a metabolic mediator and can determine the activity and function of the immune cells. In the tumor microenvironment, the combined accelerated metabolism of tumor cells and cancer-associated fibroblast creates an immunological environment that supports the tumor development. Now, in the local milieu, the tumor cells are rapidly growing. It's an anaerobic process. Therefore, they need lactic acid for their growth. Tumor tissues deplete local energy, forcing neighboring immune cells to process high concentration of metabolites like lactic acids, and in the absence of other nutrients, leading to immune suppression, and there will be increased tumor growth. Sodium lactate can inhibit several glycolytic enzymes in CD4 T cells and therefore reduce the expression of glucose flow. There will be increased accumulation of T cells at such inflammatory sites, but their activity is reduced. Level of lactic acid increases significantly as there is progression of liver disease, and it can be used as an effective tool for early HCC diagnosis as well. So these are also prognostic markers that can be used for early diagnosis of HCC. Another important metabolites are bile acids. These are endogenous steroid molecules synthesized from cholesterol, which are the main component of bile. Now we all know that bile acids can regulate differentiation and function of T cells, including T helper cells and anti-inflammatory Treg cells. Increased levels of total hepatic bile acids in HCC is seen, and this stimulates the generation of tumor-initiating stem cells. Metabolic regulator sirtuin has been reported to inhibit HCC immune escape by mediating metabolism of bile acids, and therefore a new strategy based on bile acid chelators can be used in treatment of HCC treatment. Deoxycholic acid, a secondary bile acid, usually has a role in cellular functions related to inflammation and tumorigenesis through senescence secretome, exacerbating the development of HCC. Accumulation of bile acid plays a key role in HCC development, leading to overall cell damage. Therefore, increased carcinogenesis in the presence of bile acids is mediated through various signaling pathways, and this hampers the function of the CD8 T cells, avoids recruitment of the natural killer cells, and amplifies polarization of M2 like tumor associated macrophages, therefore fostering tumor immune escape mechanisms. Now, how does the microbiota now affect this bile acid metabolism in the gut? So normally what happens is cholesterol is converted into 7-alpha hydroxycholesterol, and this is the to cholic acid and chenodeoxycholic acid, and then it is conjugated to taurocholic acid and glycocholic acid. Now this conjugation is necessary to make the bile acid hydrophilic so that it can enter into the circulation and the intestine. So once it is in the intestine, there are various bacterial groups, that is Bifidobacterium, Lactobacillus, Clostridia, Enterococcus and others, which usually mediate the deconjugation through bile salt hydrolases. So these usually deconjugate these taurocholic and glycocholic acid into cholic acid and chenodeoxycholic acid again. Certain other like Clostridia and Eubacteria, usually they express a 7-alpha dehydroxylase enzymes. And then they convert this cholic acid and chenodeoxycholic acid to deoxycholic acid and lithocholic acid. Now these two are harmful for the gut, and therefore they are implicated in the pathogenesis of HCC as well. So therefore, when there is dysbiosis in the gut, we have seen that these useful bacteria which are deconjugating the bile acids are reducing, and those which are promoting the conjugation and formation of the secondary bile acids is increasing. Therefore, this dysbiosis leads to increased presence of the toxic metabolites like secondary bile acids, which promotes the tumor formation. Thus, we see that gut microbial dysbiosis, increased gut permeability is there. Gut microbial components like LPS or the lipotecoic acid directly affect the TLR, directly activate the TLR pathway. Increased bacterial translocation is seen. Other like short chain fatty acids are altered. Also, the lithocholic acid and the deoxycholic acid usually, which are tumorigenic, causing DNA damage and enhancing the pathways leading to increased senescence associated secretome phenotype factors which are present on the hepatic stellate cells, and there is increased senescence associated secretory proteins that are released, and it leads to tumorigenesis. And they negatively affect the anti-tumor immunity by releasing prostaglandins E2. So thus, if there is dysbiosis, it is going to alter all the three mechanisms, including lactic acid, including short chain fatty acids, and bile acids. So how can we modulate that? This can be used as a therapeutic model.
Sorry, Pratibha, very nice. Can you go back? How does this from the gut goes to the HSC? What is the mechanism? You have a deoxycholic acid, but it doesn't circulate to the liver. How does it go and affect?
So basically, through portal circulation, it is going to the liver. It will be released in the liver itself. I'll read about it more, sir, that actual mechanism of the secretome pathway.
Because deoxycholic is very beneficial. It's a very good antibiotic also.
Yes, sir. But here again, it has a direct effect. I'll just show in the next slide. It's not seen. So it also causes the bile-related injury. Also is caused by the deoxycholic acid in the liver, inside the liver. So that may be a mechanism also of causing liver injury and further inflammatory responses leading to tumorigenesis, which can be seen. So how can we use this in modulating the gut microbiota? So if we increase the short chain fatty acids, it is going to prevent or treat the HCC. If you're going to increase the propionate levels or the or the interferon levels, then it is going to prevent or treat the HCC. However, modulating the gut microbiota in order that there is inhibition of the secondary bile acids in the liver sinusoidal cells, and therefore there will be reduced production of the toxic metabolites in the presence of secondary bile acids, and can prevent the HCC formation. Similarly, if Th17 cells are inhibited, angiogenic factors are reduced, there is an inhibitory effect, and it can be also helpful. So these therapies are now progressing. We'll be seeing it. Also, this was about HCC. In gut microbiota in cholangiocarcinoma is also responsible for many of the pathogenic factors. Intestinal microbiota composition is shows increased alpha diversity, increased lactobacillus, actinomyces, leptostreptococci, bifidobacterium, and increased Helicobacter species also. Now we all know that Helicobacter pylori is associated with gastric ulcers or gastric cancers, but it has an important role in cholangiocarcinomas as well, and all the species like H. pylori, Billis, and Hepaticus are related to the angiogenesis in carcinogenesis in cholangiocarcinoma. So there is overexpression of Helicobacter genes CAG-A and CAG-E, leads to phosphorylation of the proto-oncogene SRC protein kinases family, and activation of NF-kappa B proliferative signaling pathway is there in the presence of Helicobacter. So definitely there is high level of Helicobacter in the patients, in the stools of the patient who are having cholangiocarcinoma, and again it can be used as a microbial marker also for early detection of cholangiocarcinoma. Similarly, biliary infections or the pathologically altered gut bacteria, there will be increased production of LPS, and as I've already mentioned, LPS TLR binding will lead to increased pro-inflammatory cytokines and activation of the myeloid differentiating suppressor cells may be seen. Altered bile composition is also seen in the altered cholangiocarcinoma and pathogenesis, where there is increased tauroursodeoxycholic acid, increased glycodeoxycholic acid, and biliary microbiota. Bile microbiota is also altered in these patients, all leading to increased carcinogenesis. This is the list of the bacteria which are altered in the gut microbiota of cholangiocarcinoma patients. Now, this was the role of bacteria. Is there a role of fungi? Yes, because Fungi also produce certain immune responses in the presence of the pathogen-associated molecular patterns like beta-glucan or mannans, chitin and chitosan, which again activate the TLRs or NLRs. Now these specifically studies have been done where Candida albicans, which is increased in the gut of the HCC patients as compared to cirrhosis or the healthy controls. It has been shown that the nucleotide binding oligomerization domain leucine-rich repeats, that is NLRP6, is specifically associated with increased carcinogenesis associated with Candida albicans. Also, bacterial fungal interactions occur, and there is enhanced production of the metabolites. This is a synergistic effect that is seen, and it can lead to increased carcinogenesis activity here. Fungal derived metabolites are very important, and they are widely studied. These are mainly aflatoxins, farnesol, candida lysine and oxylipins. So fungal metabolites, mainly 4 types of fungal derived metabolites are there. Aflatoxins and fumonisins, these are the mycotoxins. These are seen in the food products including maize, cereals and nuts. The aflatoxins are the most common known non-infectious foodborne risk factors that has been studied in HCC, especially in sub-Saharan Africa, Eastern Asia and parts of South America. There are 4 types of toxins, of which aflatoxin B1 is the most common, and these are derived from Aspergillus flavus and Aspergillus parasiticus as a potent hepatocarcinogen, especially in patients with chronic HBV infections. Now it's very important that the population which is predominantly maize eating or having this, such cereals which are contaminated with Aspergillus flavus toxin, they have a large proportion of hepatocellular carcinomas, and also it has been shown that the populations where HBV is predominant, chronic HBV is predominant, aflatoxin has an added effect. Now fumonisins, a family of mycotoxins produced by fungi, fusarium, has also been reported to induce hepatocellular and cholangiocellular tumors. Farnesol and tyrosol, they are usually terpene derivatives of Candida, and they are act as quorum sensing molecules, and they are the virulence factors for Candida. Candida lysines, now this is a newer metabolite that is being widely studied for its cytolactic peptide toxic activity, and it is secreted by albicans predominantly, and that can damage epithelial membranes directly and activate an immune response against them. So these are also important fungal metabolites that are implicated in the carcinogenesis. We should see somewhat detail about aflatoxins, because correlation between the degree of exposure of aflatoxin B1 and the incidence of HCC is direct, with odds ratio of developing tumor of 6.37:1. So it's a very high high odds ratio, showing the correlation is very strong. Overall attributable risk is 17 to 21% in HBV positive and 8.5% in HBV negative individuals. Actually, this aflatoxin usually causes an arginine to serine mutation in the codon 249. So this 249S mutation of the p53 tumor suppressor genes is specific for the exposure to aflatoxin, and this has been seen in 64% patients with HCC. So there is a high proportion of the patients with HCC who actually have aflatoxin exposure. The mutation is present in 44% of the patients with HCC who have no evidence of cirrhosis. So in non-cirrhotic HCC as well, aflatoxin is an important hepatocarcinogen that is implicated. So these were the fungal metabolites. So now when we think of therapy for altering these metabolites, again, as I've already mentioned, what can we do to alter the gut dysbiosis? We have already seen how this gut dysbiosis is affecting through dysregulated bile or through the deconjugation of the bile acids. So various therapies have been now tried. First is FMT, regulated FMT, that is alteration in the dose or the method or the specific bacteria that are being given. Probiotics, prebiotics, diet and engineered bacteria. So they all are being tried to reduce this gut microbial dysbiosis and thereby reducing the hepatic injury. Another important is the FXR activation. Now bile acid deconjugation, which occurs in the gut, usually these secondary bile acids, they activate or they downregulate the FXR activation. So downregulation of FXR is directly proportional to the SCC generation. So if we use FXR agonist, this will regulate the alteration in the that is caused by the secondary bile acids and reduce the leaky gut barrier, and further cascade can be prevented. So these are the novel therapies that can be used based on the gut dysbiosis. mechanisms. So, future possible strategies for treatment of HCC: antibiotics like rifaximin, norfloxacin, which are capable of scavenging specific gut microbes, inhibit HCC progression. Specific probiotics, prebiotics, and postbiotics may inhibit development of HCC and maintaining microecological environment. Modified FMT, that is adjustment of the components, delivery frequency, delivery route, can bring the gut microbiota to the normal, closer to the normal people, and it can revert the changes. And the immunosuppressive environment shaped by harmful species of the gut microbiota can be improved by immunotherapy using the anti-CTLA4 or the PD-1 inhibitors. FXR agonists such as the obeticholic acid and TLR4 inhibitors such as erituran can affect the bile acid metabolism and further immune response signaling, which may be beneficial. So, this was about how the dysbiosis is affecting the pathogenesis of HCC. Now, do these bacteria also cause infections in HCC? Because that is what we are going to treat and we are going to detect, right? So, patients with HCC or cholangiocarcinoma are prone to various types of infections, either due to the liver dysfunction, immunosuppression, invasive nature of the diagnostics and therapeutic procedures. Most common are the bacterial infections, and most common among them is the formation of liver abscesses. It is secondary to the bacterial translocation or the secondary to the procedures such as biopsies or TACE, and most commonly implicated bacteria are E. coli, Klebsiella pneumoniae, and Staphylococcus aureus. Other important infections are cholangitis, again inflammation may be due to obstruction or procedures. E. coli, Enterococcus species, and Klebsiella are most commonly implicated. Spontaneous bacterial peritonitis can also occur in these patients, and usually it is because of Enterobacteriaceae family or also because of Streptococcus species. Pneumonia, though a rare complication, it's usually associated with the hospital stay and associated with the hospital acquired pathogens. Bacteremias and sepsis can occur in these patients and leading to multi-organ failures. Viral infections, there can be reactivation of HBV, HCV, and other viruses like HSV, varicella zoster virus, or CMV, owing to the immunosuppression in such patients. Fungal infections, I've already mentioned. Candida infections, particularly in immunosuppressed or those undergoing procedures, will be increased. Aspergillosis have also been reported, especially after therapies for HCC. And Cryptococcus, particularly in patients with advanced liver disease and immunosuppression, have been noted. There is also case reports of amoebic liver abscess and schistosomiasis leading to chronic infection which can cause hepatic fibrosis and cirrhosis, and this may complicate actually the picture of HCC. Other nosocomial infections like catheter-associated infections and surgical site infections can occur. Now, infections in cholangiocarcinoma, are they increased due to the colonization in the bile? So, there was one study where they have studied that the bile colonization predisposes to post-op biliary tract infections. They have studied a cohort of 95 prehepatic cholangiocarcinomas patients, and who underwent surgery between this 9 years period, and their intra-bile cultures were available, and they analyzed the bile duct colonization and then the post-operative infections as well. Now, what they found was that 84.2% showed bacterial colonization of the bile duct, so their bile cultures were positive, and subsequently 54.7% they developed the post-op abdominal infections. Enterococcus fecalis, faecium, and Enterobacter cloacae, E. coli were the most common colonizing bacteria. And subsequently, the infections were also caused by Enterococcus faecium, Enterococcus fecalis, Enterobacter, and E. coli. Therefore, the bile colonization is also very important. The bile microbiome is also very important to for the development of the subsequent infections in such patients. Similarly, for prevalence of HCC, the infections, as I've already mentioned, there can be presence of SBP in these patients as high as 20% of the cases. And again, the Enterobacteriaceae family is predominantly involved, E. coli, Klebsiella, and even Pseudomonas species was found. Gram positives, mainly Enterococcus and Staphylococcus, were the predominant organisms. What important here is that patients with SBP, having SBP without HCC, they had good response to therapy, as well as 30-day mortality was less. So, there was a higher 30-day mortality in patients with HCC, and also initial response to treatment was poor in case of patients with HCC who had SBP. Now, patients who are undergoing therapies for HCC may also develop infections, especially.
Summarize,
yes, sir.
Please. Please.
SBRT associated infections. Post-ACE infections are important because there have been increased incidence of post-ACE infections ranging from 0.1 to 4.6%, and however high mortality is seen if there are such infections up to 50% of the cases. TACE was associated with a higher risk of developing symptomatic bacterial infections than the hepatic arteriographies in one of the studies, and previous gastrectomy was possible risk factor for liver abscess formation following TACE. So there are many reports and studies showing increased incidence of infections following TACE. However, PCT is a diagnostic factor that can help in predicting the prognosis of patients with unresectable HCC which are treated with TACE. So coming on to ILBS data, I have analyzed the 2000 patients who had hepatobiliary malignancies. This is the combined data, and the culture was positive in 40% of the cases. It was only one type of infection which was considered. We had positive cultures in 43%. Most common was urinary tract infections, followed by body fluids including ascitic fluid or pus, liver abscess pus, or the PTBDs. And others with the 15% had blood culture positive, and respiratory infections were lower in number. In the blood cultures, it was gram-negative enterobacteriaceae which was predominant. Also, polymicrobial infections were also high in such patients. So polymicrobial infections are actually more common in HCC and cholangiocarcinomas. So they were also reported. And others like Brucella and Candida were also isolated. Body fluids, we can again see the gram-negative bacteria predominated, along with the polymicrobial flora was there, and fungal and other gram-positive cocci were also seen in lesser numbers. Urine cultures, predominant it was the gram-negative bacteria, followed by gram-positive bacteria. However, large proportions were polymicrobial, and 10% of them were fungal, predominantly Candida infections. Now, distribution of the organisms, as you can see, Klebsiella pneumoniae was predominant in our setting, followed by E. coli. So Enterobacteriaceae was predominant, followed by Pseudomonas. Pseudomonas again a hospital acquired pathogen. Few other cases like Enterococcus, Staph and Salmonella were also isolated. If we see the sensitivity pattern, again it's a very poor sensitivity that these organisms show to most of the antibiotics, and only the second line antibiotics had a good response in this bacteria. Sensitivity pattern of the GPCs, again this is also poor. We can see these are the resistant ones, red ones, which are higher, and even vancomycin, especially in Enterococcus, vancomycin resistant Enterococcus, linezolid resistant Enterococcus were seen. Sensitivity for Candida species, there was a reduced sensitivity to fluconazole as compared to the other agents. However, others like voriconazole, caspofungin and amphotericin had good sensitivity. So when we compared these 2000 patients who had no infection and infection, we could see that the univariate analysis showed that there was a significant difference in hemoglobin, the platelet count, eosinophil levels, INR, creatinine, bilirubin, and also serum SGPT, serum chloride levels and sodium levels. Mortality was significantly higher in the patients who had infections. It was 17% as compared to 3% in the no infection group. Logistic regression analysis showed that only serum SGPT, TLC, total bilirubin, eosinophil and serum sodium were independent factors contributing to the development of infections. We had also published this paper, association of Streptococcus gallolyticus subspecies pasteurinatus in HCC. So we found that when this pathogen is isolated both in ascitic fluid and in blood, it is significantly it can be a prognostic marker of developing prior to the actual clinical signs and symptoms of HCC. So way forward, we have to work on gut modulation therapies for hepatobiliary malignancies, FMT for HCC and cholangiocarcinomas, and aflatoxin testing can be done. Thank you. I would like to thank Dr. Gayatri, Dr. Archana, Gure sir and Dr. Samruddhi. Thank you, sir.